Welcome to the recent edition of cube-biotech Scientific Masterclass series, where researchers from around the world present their latest results and findings to a global audience. As I said before, my name is Michael Erkelens, and I'm part of the research and development team of cube-biotech. Today, I'm happy to introduce this masterclass series, and this masterclass series should provide a unique platform for scientists to share their work and to connect with others in their field and receive feedback from experts and peers. Maybe at the beginning, a little organizing stuff. So after the talk today, there will be time for the interactive question and answer series. So during the talk, you will be able to use the chat function on the bottom right of your browser. And with this chat function, you can comment directly aspects of the talk, or you can place a question for the Q&A session afterwards. If you use the button with the question mark in the chat function, you can directly place a question, and then afterwards we can highlight the question on the desktop, and our speaker can directly answer to the question. Well, so our first speaker is Dr. Mario Leutert, and he's a postdoctoral researcher at the ETH Zurich. And for me, it is a great pleasure today to introduce him and also his CV. So first of all, hello to Dr. Leutert. He was born in 1989, and he started his early academic career in 2009 at the ETH Zurich. So in the Department of the Biosystems, Science and Engineering. And in 2014, he graduated in Harvard. Afterwards, he came back to Zurich and obtained his PhD in the group of Professor Dr. Michael Hotteger. During his early postdoctoral career at the University of Washington, he won multiple publication awards for his publications in high-ranked journals, so for example, Nature Communication. Additionally, he was awarded with the Early Career Research Award of the US Human Proteome Organization. At the moment, he is a postdoctoral fellow in the group of Judith Wellen at the University of Washington and in the group of Pedro Beltrao at the ETH Zurich. Mostly, his research is focused on developing and applying computational mass spectrometry-based proteomic technologies. But I think he will give us a detailed overview in his science in a few seconds, I think. So again, hello, Dr. Leutert, and maybe you can turn on your camera so the people can see you. Nice. So hello again. And did I forgot something? So would you like to add something to your CV, or was it comprehensive?
Yeah, no, that sounds great. And yeah, thanks a lot for providing this exciting platform to present my research here.
Yeah, it's a pleasure for us. So your recently published paper is a nice example for the emerging field of proteomics, and especially of the importance for the aging process and so on. So I'm really a pleasure that you will give a talk and an overview over your paper. Yeah, so then maybe I think let's start. You can open your presentation and I will give you the word.
All right, so you see my presentation?
Yeah, we can see your presentation. Great.
Okay. Okay. Well, yeah, thanks everyone for attending my seminar, and thanks again to cube-biotech for providing this platform. So I'm going to talk today about multidimensional proteomics and how we use it to decipher regulatory protein networks in cellular signaling and aging. Yeah, so first I'll give you an overview what actually is multidimensional proteomics and why we think we need it. I'll show you how we can implement it, and I'll show you how does it help us to reveal biological functions of protein networks. And for this, I'll give you two examples. First, how we study kinase signaling networks, and second, how we study protein complexes in cellular aging. So with the proteome, what we mean is all the proteins that exist at a given time in a cell. However, the proteome is a function of interconnected and dynamic protein properties. So these protein properties or dimensions are of chemical, physical, and biological nature. So for example, that means structural properties of proteins, but also their solubility, their activity, but also where they're localized in a cell, with what other proteins they're interacting, what post-translational modifications they carry, obviously their abundance, but also their turnover, so their synthesis and degradation rates. And it's a very exciting time right now because we can actually measure many of these proteomic dimensions using mass spectrometry-based proteomics. However, if you have a given proteome and you measure, for example, protein abundance, turnover, and phosphorylation, it doesn't really tell you anything about the functions of the proteins within this proteome or what the proteome actually does. To do that, you would have to integrate across multiple cellular states. So, for example, across different perturbations, across different time points, or different cell types. And obviously, this gets very complicated, and it's often impractical or impossible or even unnecessary to measure that many conditions. However, if you choose the right combination of proteomic dimensions to measure, that can give you very deep insights into biological mechanisms. And how you choose your path through this maze basically really depends on what biological question you want to ask. So, for example, we are interested in signaling networks, so kinase-mediated signaling networks. And one way how I showed you today, how we did this is we actually only looked at protein phosphorylation, proteome-wide, but we did this for a couple of hundred different perturbations. And in another example that I'm going to show, we were interested in how protein complexes are affected by cellular age. And in this example, we only looked at a few different time points. However, we acquired 4 different proteomic dimensions, so abundance, turnover, phosphorylation, and stability. In any case, if you do multidimensional proteomics, you often end up with a lot of different samples that you need to acquire on your mass spectrometer. And this is not straightforward because proteomics is still limited in throughput and reproducibility. And the reason for this is that the sample preparation of proteomic samples is quite involved. So you have to lyse your cells or tissues, you have to extract the proteins, you then have to digest these proteins into peptides, you have to perform some kind of purification. And then if you're interested in post-translational modifications, you also have to do an enrichment at the peptide level, and only then can you perform your mass spectrometry analysis. And an obvious solution to this problem would be to perform the sample preparation in a 96-well format, to streamline it and to automate it. And also downstream on the analytical side, there are some considerations that you need to make to acquire that many samples. However, today I'll only present to you our solution for the sample preparation step here. And basically what we wanted was a method where we can use cell lysate in a 96-well plate format, use some robotic automated method, and end up with peptides that are ready for mass spectrometry injection to do a whole proteome analysis. And then in a second step, also phosphoproteomic sample preparation. The method we came up with, we call it R2P2, or Rapid Robotic Phosphoproteomics. And basically the idea of the main principle of the method is that in the last few years, basically all steps in proteomic sample preparation have been shown that they can actually be performed using paramagnetic beads. And so in our approach, we use a paramagnetic bead or particle processing robot, so a Kingfisher from Thermo Scientific. And this robot is basically depicted here. It has 8 slots where you can put in 96 well plates. And then the robot has a 96 well plate head with 96 magnetic pins. And what it can do, it can move magnetic beads across 96 well plates. Basically, it can capture the beads, it can release the beads, it can agitate the beads, it can heat the beads up, and so on. And so we implemented a method based on the SP3 methodology. And how this works, I'll just walk you through really quickly, is we have our cell lysates in a 96 well plate format. And we bring it to a high organic concentration, so usually ethanol up to 80%. And what now happens if you mix your cell lysates under these high organic conditions with the magnetic particles is that the proteins fall out of solution and they aggregate on the magnetic beads. And now the robot basically moves these bead protein complexes across different 96 well plates. They all contain high organic solvents, and thereby you get rid of salt, detergent, lipids, so everything that you don't want in your sample. And so you end up with a highly pure protein samples that is then mixed with some protease, so trypsin or lysine. You digest the proteins into peptides. You then separate the magnetic beads from the peptides, and your peptides are clean and ready for mass spectrometry analysis. And then if you're also interested in phosphoproteomics, you can do a second robotic run. And there you take your 96 well plate with the peptides, you bring it again to a high organic concentration under acidic conditions. You mix it with iron IMAC beads, and then the robot moves this again across different 96 well plates with organic solvents. You elute with base, and you end up with highly pure phosphopeptides that can again be injected into the mass spectrometer for phosphoproteomic analysis. So this method is published. You can find all the details and protocol in here. And just very exciting, a couple of days ago, Alexis Chang from our lab, she published an additional protocol that you can basically couple to this method here. And this allows you now to also specifically enrich for phosphotyrosine peptides. So that also shows that the method is really modular. Okay. So now having this really high throughput and reproducible method in hand, I was interested to see if we can now use high dimensional phosphoproteomics to get insights into signaling networks. And I'll show you an example from a study that we published just at the end of last year. And there, what we did is we wanted to generate a really high dimensional atlas of phosphoproteomic stress responses in the budding yeast Saccharomyces cerevisiae. And so we exposed this yeast to all kinds of environmental, chemical, and drug perturbations. So overall, around 100 different perturbations. All of them were really short-term, so mostly 5-minute treatments that included, for example, carbon and nitrogen source starvation, cell wall perturbations, DNA damage, drug treatments, and changes in the pH of the media. And the goal here was to really engage the whole phosphosignaling networks in all way possible. So provoke every phosphoproteomic response that you could imagine. And then we did this in 6 biological replicates. And for each sample, so resulting in around 700 samples, we processed that with our R2P2 method. So overall was around 7 different robotic runs. And ended up measuring the phosphoproteome of each individual sample. And so that's basically how it looks like. So we created one of the largest perturbation phosphoproteomic data sets. Here it's listed by perturbation type, and each bar is the amount of unique phosphosite that was identified in an individual perturbation. And we see we have around 10 to 15,000 unique phosphorylation sites, and around 5,000 of them are reproducibly measured across all samples. So obviously what we can do is compare to our untreated conditions. So how many phosphosites are regulated in the individual perturbations, and that's depicted here as a bar plot. And we see already here that for the different perturbation types, there's quite some structure in this regulation. So for example, a lot of the nutrient perturbation provoke a really pronounced phosphoproteomic response, whereas other perturbations only have kind of selective responses. But importantly, we measured around 25,000 regulated perturbation phosphosite pairs. So now in your normal phosphoproteomic study, what people would do, they just take one perturbation, compare it to the untreated, and try to figure out what's going on. But here, because you have such a high dimensional data set, we can follow a completely different approach. And I'll show you one example of this here. So maybe you've seen such a type of plot before. It's called a UMAP plot. It's very common in high dimensional data sets such as single cell transcriptomics, but let's just walk you through it really quickly. So each individual dot here is an individual phosphorylation site. And the underlying data is how is this phosphosite regulated across the 700 samples. And so it's a very complex high dimensional data set. And you apply this UMAP approach, which is somewhat comparable to a principal component analysis, and you bring it all down in the same two-dimensional space. And now it's easier to analyze them all at the same time. And so now if phosphosites end up in close proximity in this UMAP space, that means they're regulated very similarly across the 700 samples. And what does that now mean for a phosphosite if it's regulated in a similar way across all of these samples? It probably means that it's either that this phosphosite is either modified by the same kinase, it's in the same signaling pathway, or it carries out a similar function in the end. And we already get a hint at that this is true when we color our phosphosites here by very broad motif types. And a motif, that's basically just the immediate amino acid surrounding of the phosphosite. And often kinases, they need to recognize the surrounding of the phosphosite in order to be able to put the modification there. And so we see, for example, a lot of phosphosites here in the middle of this structure are in a basic motif. We have acidic motifs over here, or really proline-directed phosphosites over here. But obviously, we can look at this in a much more detailed way. And for this, we applied some clustering algorithm and identified modules of phosphosites here in the different colors that are regulated in a similar way across all of the perturbations. And then we can take these modules. The modules have around 20 to 150 phosphosite inside, and try to figure out what are these signaling modules. And just show you an example here for 3 of the modules. So first we can look how are these modules regulated, these like phosphosite groups regulated across perturbation. So for example, module A, these phosphosites are downregulated in osmotic stresses and in starvation conditions. Module B is downregulated in temperature shock and cell cycle perturbation, and phosphosites in module C are highly upregulated in osmotic stresses and heat shock. Then additional piece of information, we can do a motif enrichment analysis, so more detailed as before. And in this plot, basically at position 0 would be the phosphosite, and then we see what amino acids are enriched in the immediate surrounding. Then module A, for example, has these 2 characteristic arginine sites here, and that's very typical for AMPK or TOR-related kinase. Module B is in a very acidic patch, so that's usually common for casein kinases. And then module C, these are proline-directed phosphosites, and that's very typical for MAP kinases. So we add already an additional layer of information. And then we can actually analyze where are these proteins localized that are getting modified in these modules inside the cell. Module A is cytoplasmic, module B is in the nucleus, and module C is specifically on the cytoskeleton. And then as the last piece of information, we map the phosphoproteins that are modified in this module on the global protein-protein interaction network. And you can already see these phosphosites are in close in proximity within the interactome. And we also plot some known pathway targets within this within these modules. And you see, for example, module A has quite some TOR-related kinases inside, and module C, for example, targets that are already known MAP kinase targets. And now, obviously, the really exciting part about this are all these black proteins, because they've never before been associated with these specific kinases or pathways. And so we think by doing this analysis, we really kind of bring in some organization in the global phosphoproteome that we have measured, and kind of classify phosphosites to which pathways they belong. And yeah, that was just a quick example. And yeah, I think we can use high-dimensional phosphoproteomics to reveal functional organization across the kinase signaling network in a pretty unbiased way. Okay, and now I'll show you the second example. This was from a study that we published on bioRxiv just a couple of weeks ago. And there we wanted to know how can we use multidimensional proteomics to study very complex biological processes, such as, for example, aging. And again, we used yeast, Saccharomyces cerevisiae, as a model system, this time to study aging at the systems level. And so cellular aging is obviously a really important process. All of us age, and we don't get healthier when we age. So it's important to study. But also, cellular aging is very complex because it's a multifactorial process. And eventually, that can result in declining functions of the proteome. But it's not really clear which functions are declining. And then just really quick introduction in the aging model in yeast. So we study replicative aging in yeast. And so yeast cells, they divide asymmetrically. So you have the yeast mother here, and it generates daughter cells. And the yeast mother cell can produce around 20 to 30 daughter cells, and then it dies, basically, of age reasons. And very nice, the exact age of the mother is measurable because every time it produces a daughter cell, it creates a bud scar. And then very interestingly as well, all of these daughters, they are rejuvenated. So even daughters from old mother cells, they have, again, the complete lifespan or lifespan potential. And then this system has similarities to asymmetrically dividing cells of higher eukaryotes, such as, for example, stem cells. And a lot of aging pathways in yeast have been found to be conserved also in higher eukaryotes. And so what we did, we measured protein abundance, turnover, stability, and phosphorylation across the age of yeast mothers as well as their daughters. And basically now model how do these proteomic dimensions change as a function of age. And our aim is to find out how our proteome functions impacted by cellular age. And this is just an initial overview. So you see this is comparing young, middle-aged, and old mothers versus the initial daughter cell for the different proteomic dimensions. And you see there's increasing proteomic features are changing with increasing age. So there's really a lot of proteomic remodeling going on upon aging, but also we see this for all of these different proteomic dimensions, so indicating that these are really relevant dimensions to study upon cellular aging. And I just give you one example for where we could confirm an aging hallmark in yeast, and this is the increase in vacuolar size. So here, these are our yeast cells, and you see, for example, this is a yeast cell, yeast mother of age 4, so it produced already 4 bud scars, whereas these ones have an increased age. And one hallmark of aging in yeast is increase in vacuolar size, and you see that here. So the older the cells get, the higher the vacuolar load, whereas in the daughter cells or in the buds, this doesn't change. And we can validate this by looking at the protein abundance dimension in our data. So here, as a change versus the young mother, we see the older the cells get, the higher the load of vacuolar proteins, whereas this doesn't really change in the daughters, even of the old mothers. Okay, but now the thing that we found really interesting is actually kind of aggregating this and analyzing at the level of protein complexes. How does this age-mediated regulation look like? And so what I show here is for the different proteomic dimensions, so protein abundance, turnover, and stability, how protein complexes are affected by age. So in this plot here, we show on the y-axis is basically an age score. So the higher the score, the more a protein feature increases during aging. And if it's a low value, that means it's decreasing upon cellular aging. Each dot is a protein complex, and it's a ranked plot, so everything over here really increases a lot upon aging, and stuff over here decreases upon aging. And so we show this for the cytosolic ribosome, the proteasome, and the electron transport chain. And now already just looking at these three protein complex classes, you see they're completely differently regulated across cellular age. So that's already really interesting. So for example, electron transport chain seems to increase in abundance during aging, whereas the cytosolic ribosome is really decreasing in abundance. But then also you can look at the turnover dimension, and there the cytosolic ribosome has a really high turnover. And so if you now kind of bring this together, you can already make mechanistic conclusions. So for example, cytosolic ribosome seems to be actively degraded, and that might be why it's the very low abundance in the old cells. You can also look at the stability. So the cytosolic ribosome is destabilized, and so is the proteasome. And for a protein complex, if it's destabilized, that often means it doesn't form the proper complex anymore. Yeah, so I'll just give you two more examples how we can dive even deeper into this. So if we now look at the proteasome, so we already have all of this information, but if we kind of look across all the age points, how the stability changes, we see that the lid of the proteasome is not really affected by cellular age, but then the proteasome core is really destabilized. So you probably end up with a lot of non-functional proteasomes in old cells. And how can we explain this? We can go again back to the protein abundance and turnover data and basically calculate loss of stochiometry in the complex. So meaning how are the subunits of the complex regulated compared to the average abundance or turnover of this complex. And what we see there is that indeed the proteasome core with a really high loss in stochiometry. So that might explain why this complex is destabilized. So it just doesn't have the right ratio of subunits to form the proper complex. And if we take this a step further, we can also look in the daughters of these models, how does the stochiometry of the complex look like? And there, very interestingly, somehow the integrity of the proteasome or the stochiometry is preserved in the daughters. And so that's probably really important also for the fact that these daughters are rejuvenated and have again a full lifespan potential. And yeah, we can basically do this for all of the protein complexes that we measure with a high coverage, and then we can create these little models, mechanistic models, and predictions how the protein complexes are affected by cellular age. And as I just showed you for the proteasome, we saw that it's not actively degraded. However, you have a loss in stochiometry, and this might explain why the whole complex is destabilized and why probably you have less functional proteasomes in old cells. Okay, so as a summary, I showed you that robust and cheap high-throughput proteomic methods are now available, and they really open up a lot of new opportunities to study biology in a way that we were not able to do before. I've also shown you that high-dimensional phosphoproteomics can reveal functional organization principles of kinase signaling, and then multidimensional proteomics can give you deep insights into age-affected processes. So, for example, how protein complexes behave upon increased cellular age. And yeah, with this, I come to my acknowledgement. So all of this work has been done in the lab of Judith Wien, and I got a lot of help of my lab mates and my collaborators. And now I'm in the lab of Pedro Beltrao at ETH Zurich. And I'm happy to take any questions. And if you have questions later, it's also pretty easy to find me online. Thanks.
Nice. So thank you very much, Dr. Leutert. So it was a great talk, and I really like the holistic view, so which you generate through your new technique. And maybe you allow me to ask the first question. So also because of your holistic view, are there other techniques necessary to yeah, let's say yeah, so give additional information about the proteins, because, for example, if you look at the phosphoproteomics, so then you need maybe immunoprecipitation or different IMAX, so with different ions. So what do you think? Is it the unspecific aggregation of the proteins on the beads? Is this enough to give the holistic view?
Yeah, I mean, I'm a really big fan of combining different angles and different views to, as I just showed you for the aging project, to get really deep insights. So yeah, often if you have more information, you can also make better conclusions. But then it often becomes the question, I mean, is it really practical, or do you have the resources to like measure more and more and more? Because yeah, at some point it's the resources you need don't really justify the output that you get. So often it's like you have to think before and design the optimal experiment, and that's usually the best way.
Nice. Thank you very much. So let's come to the official F&A mode with a question and answer mode. So I will change to the F&A mode. And now let's have a look. There are several questions. The first question from Toman Borgtikin. He's curious about the protein peptide input range of the R2P2 protocol. So what is the minimum amount of peptide input used for the phosphopeptide enrichment? Is there an upper limit to how much protein can aggregate with the beads in the initial enrichment step?
Okay, yeah, so maybe, yeah, I'll first answer about the upper limit. So in theory, based on the method itself, on the SP3 method, there is no upper limit. However, it means when you process a higher amount of proteins, you also need a higher volume. And so when we do this method on our robot, we're basically limited by the volume like each well in a 96 well plate can take. And so I think for 96 well plate, that's 1 ml. And so usually we do the protocol at 5 mg per ml. So yeah, if you, let's say, if you have to process more than 1 milligram or 5 milligrams, it's much easier to do it manually. So usually a really good range is like 250 micrograms. That's what I usually use for the limit at the lower end. Yeah, I mean, it works perfectly fine if you have 25 micrograms. If you go much lower than that, then yeah, probably depends on your sample type, and you might have to do some optimization. But in general, people have shown that the SP3 method, so this magnetic bead method, is really sensitive, and you can do kind of low input proteomics.
Okay, so if there is an additional question to this, so just you can also ask another question, and we will ask it in detail. Well, so the next question is from Bernd Kühnemann, and Bernd Kühnemann asks, what kind of phospho enrichment residues can you use in your automated protocol?
So basically, you can use any residue you want as long as it's on a magnetic bead. And the only thing is you have to kind of optimize or use the suggested organic solvents for the enrichments and for the washing of the beads. But yeah, we have done it with titanium dioxide, with titanium IMAC, with zirconium IMAC. Yeah, with basically a lot of different things. So yeah, you can choose whatever you like and just use the suggested protocol for that.
Okay, so the next question is from Peter Darrow. So Peter Darrow is curious about how or even you can transfer these results from Cerevisiae to another model organism or to humans.
Yeah, so to transfer it, I mean, not the easiest way, but one way is there's a lot of conserved pathways in humans that we've already observed in yeast. It's often the problem is then if you go to human, these pathways are expanded, so you have more kinases, more substrate, and so on. But yeah, you can make some predictions kind of how the pathways are regulated and transfer this to another model system. And yeah, so that's basically that. Yeah.
Okay, nice. So the next question is from Carmen Mejia. She asks, could you tell me what future potential you see in phosphoproteomics?
So I think phosphoproteomics, on one hand, we're really lucky. So the methods and the sample preparation is really advanced. So you can get extremely highly pure phosphopeptide samples. You can get very deep, like measure in human cells, like 30, 40,000 phosphosites easily and get very good coverage. You can do very good quantifications. But that said, the really big challenge is actually more downstream. So how do you make sense of the data and kind of the bioinformatic tools they use? So first of all, yeah, you need to be able to use this type of tools. It's often customized tools. But on the other hand, it's also phosphoproteomics, so phosphosites, only very few of them actually have an annotated function. So I think it's maybe 2% of all phosphosites, we know the function of it. So for the other phosphosites, yeah, you need to find other ways how to understand it. And so now a really promising approach is to kind of combine phosphoproteomics with other functional proteomic assays. So for example, thermal stability or turnover, and that can give us more hints which phosphosites are actually functional.
See, the next question is asked by Philip Harnisch, our head of biology from cube-biotech, and he is asking, could you imagine using this specific method one day in human medicine, for example, identifying specific key players in different human sicknesses, or checking which parameters are up and down regulated to adjust the therapy on a cellular level?
Yeah, I mean, these methods readily can be applied to human cells or disease models. And I think now the methods I presented, these are really kind of discovery methods. So yeah, they might help you to identify disease mechanisms and so on. And then maybe if you identify an interesting protein or interesting phosphosites, you can basically transfer this to more targeted methods, which then might find some application in biomarker monitoring in the clinics or something like that.
So it would be also useful for drug screening and stuff like this, all right?
Yeah, exactly. So all kind of discovery-based projects is, yeah, it's really nice.
Nice. So the next question is from Jeremy Thorner. Jeremy Thorner is asking, for the yeast kinase network analysis, did you look at any kinase mutants as well as wild type cells to confirm your conclusions?
So I did some comparisons with kind of really wild yeast isolates, and yeah, that's interesting. So that can give us information about the evolution of kinases. In terms of kinase mutants, so I personally have not done it, but there's a really large-scale analysis from the Steve Gigi lab at Harvard, where they systematically acquired phosphoproteomes for all different types of yeast kinase mutants. And it's a very nice data set, and I've used it a lot to analyze my phosphorylation data. But yeah, this is kind of that goes into the direction. It's hard, and we need to be much better at identifying which kinase is actually modifying which substrates.
The next question is asked by Emma Turner. And Emma asks, how much can the preservation of proteasome integrity in daughters improve the replicative age if 20 to 30 buds before death are considered as average?
Yeah, so I mean, people have found that improving proteasome integrity can actually prolong lifespan. So people, how they found it is they upregulate the transcription factor that is responsible for transcribing the proteasomal genes, and I don't have the data in mind, but I would guess it's maybe 20 to 30% increase in lifespan or something like that. So yeah, quite significant.
Okay. Nice. So I think there is 1 minute left, so I will ask maybe the last question. So personal, what is the most exciting development in proteomics for you in the last years? So I think it's an emerging field, and in the last decade there are lots of investigations. So, but what is your favorite in the last years?
Yeah, so I think we became really good at the instrumentation and actually measuring protein abundance or post-translational modification states and so on. So for me, the most exciting part is now where people take this proteomic technology and actually combine it with cell biology methods. So for example, as I showed you, protein stability measurements or turnover measurements or proximity labeling measurements. So it's kind of this combination can give us really deep insights to in vivo how our cells really working and how reveal different mechanisms.
Okay. Oh, nice. Yeah, this sounds great and promising. So, the other questions, we will answer the questions, and so for the asking people, we will answer the questions and stay in contact with you, and then you get the answer by mail later on, because we're running out of time. And so I would like to again thank you very much for your talk. It was a great talk, and your research is really, really holistic. And I think this is where the key research is done, right? The holistic view. In the past knockouts and so on give only a little insight, and this is a great technique. So I'm hyped.